Everyone, I'm Anusha, and today we're going to look at the factors, we're looking to look at enzymes and certain factors that affect their activity. So, just before we go into those factors that affect the enzyme action, let's just take a look at enzymes. A quick recap. So, in this chapter on enzymes, we have to remember that enzymes are proteins, and remember there's different levels of organization of the protein: primary structure, secondary, tertiary, and quaternary structure. So enzymes are globular proteins, and remember, with globular proteins, tertiary structure is very important. That's because enzymes have a very specific site called the active site, which is complementary to the substrate. And in order for the active site to have this very specific structure shape, the tertiary structure is there that gives it this shape. So the way the polypeptide chain folds itself to give this structure is very important. So obviously there's bonds also that we need to remember. So bonds such as the ionic bonds, covalent, so disulfide bridges, the hydrophobic interactions, which are not really bonds, they're just the way the R groups come together to exclude water, or they may project into water. And with the hydrogen bonds as well, that we should not forget. So, so in this picture here, if you watch my cursor, you can see that the substrates have a complementary shape to the active site. And so using this term complementary is very important when we are explaining enzyme action. So we see the substrates bind to the active site to form an enzyme substrate complex. So that's also very important to mention. And finally, we have the enzyme product complex, and then the product is released from the enzyme. So when we're explaining enzyme action, so it's important also to mention how the enzymes catalyze the reaction. So there's the term activation energy that is important to mention. So what's activation energy? Activation energy is the energy required for a reaction to happen. So we know in the body, so body temperature is quite low and reactions may be very slow without the enzymes. This is why enzymes are needed to speed them up. If you look at the energy profile diagram over here, we can see that this reaction here of glucose with oxygen, we can see how much activation energy would be required without the enzyme. And with enzyme, this is so little activation energy there that is needed. So that's what enzymes do. They catalyze reactions by reducing the activation energy that is needed for the reaction to happen. And yes, so we mentioned the active site, we mentioned enzyme substrate complexes, we mentioned the reduction of activation energy. Another point that is worthwhile understanding is that enzymes are very, very specific. And the specificity is because of the very precise shape of the active site that is very complementary to the substrate. So we've got so many different types of enzymes that catalyze different reactions. Okay, so so just a quick recap here about the protein tertiary structure that we mentioned is very important for the enzyme's precise active site. So this is folding the polypeptide chain, and this is driven by 4 kinds of interactions that we mentioned. So there's the R group of the amino acids are to do with this precise folding of the chain upon itself to form a precise 3-dimensional shape. Okay, so now let us look at two models that are important to explain enzyme action. There's the lock and key model, and there's the induced fit model. So, very briefly, the lock and key model, it's to do with a lock and a key. So the enzyme being the lock and the substrate representing the key. And we know that locks have a very specific shape and only one kind of key will fit in the lock. In the same way, our enzyme, according to the model, so the substrate, according to the model, fits exactly into the active site of the enzyme to form the enzyme substrate complex. So the lock and key complex here, that's the analogy. And then we have another model called the induced fit model, which states that initially, unlike the lock and key model, the substrate shape, the shape of the active site is not quite complementary to the substrate. So you can see that here from the diagram. But as the substrate fits onto the active site, the shape of the active site changes as the active site molds itself around the active site molds itself around the substrate. And so then it becomes complementary to the substrate, although initially it wasn't. So this is the induced fit model. It's a bit like a glove. So we know that the glove initially is not the shape of our hand, but once we slip our hand into the glove, then the glove molds itself around our hands. So that's the induced fit is like that. So when we're choosing which model to use to explain enzyme action, we have to think of what kind of metabolic reaction it is. Is it an anabolic reaction we're explaining or a catabolic reaction? So let's see. So actually, the lock and key model is a model that's more suited for anabolic reactions, whereas the induced fit model would be more suited for catabolic reactions. So just very briefly, just to explain what anabolic and catabolic is about. So we have lots of metabolic reactions taking place in our body, in our cell at any one point. Some of these metabolic activities would be catabolic activities. So if we look here, so catabolic, one way to remember is the word cut. So when we cut something, it's a complex, something complex that we break up into little bits. So in a catabolic reaction, it's breakdown reactions, whereas anabolic reactions would be build-up reactions. If we look at this picture, here we can see two substrate molecules fitting into the active site, and they then become so that there's bonds formed between them, and then you end up with one new compound. So two substrates into one, so that's a build-up. It's a synthesis, a build-up reaction. So that would be an anabolic reaction. Over here is a breakdown reaction. So you can see a complex compound fitting into the active site, and then after the reaction, you got to small products. So this is a breakdown reaction. This is a catabolic reaction. So examples will be, for instance, the formation of maltose from starch. We know starch is a complex carbohydrate when it's digested into maltose. So the maltose would be smaller, it will be a breakdown product. So the formation of maltose from starch would be considered a catabolic reaction, whereas an anabolic reaction would be, for instance, synthesis of starch from glucose. So we're starting with simple compound, simple molecules, and we're getting a bigger, more complex compound. So, yes, so let's just take a look at the previous slides. So we'll come to anabolic reactions and catabolic reactions in a minute. So yes, okay, let's do it now. So how do we explain enzyme action for catabolic reactions? So it's important to mention points, especially if we're sitting an exam and we want to earn marks. So we will use the correct terminology as far as possible. So we'll say enzyme and substrate collide to form an enzyme substrate complex. Catalysis occurs at the active site of the enzyme, and we will say what model. So for catabolic reactions, the induced fit theory is better. Why? We're going to explain now, because so initially the active site is not quite complementary to the substrate, but then as the substrate fits in, there is a conformational change in the active site, and that distorts the substrate molecule. And this distortion is what lowers the activation energy and allows bonds to break. So here's a breakdown. So catabolic reactions break down, we're breaking bonds, and this distortion allows this. So we are going, therefore, for catabolic reactions for the induced fit theory. It's more suited. How about anabolic reactions? So the enzyme and substrate collide to form an enzyme substrate complex. Catalysis occurs at the active site with a lock and key model. So it's always good to explain what the model is about. So the active site of the enzyme is complementary to the substrate, which fits exactly into the active site. Substrate molecules. So how are build-up reactions facilitated by enzymes is that substrate molecules are held, and they are held in such a way, they're orientated in the right position to facilitate bond formation between them. In this way, the enzyme lowers the activation energy for the reaction. So we know that molecules are constantly moving. They're moving, they're vibrating, rotating, and they're moving about. And in order for a reaction to happen, the enzyme and the substrate molecules, which are moving about randomly, need to collide successfully to form the enzyme substrate complex, which we had discussed before. So having said that, in order for bond formation to happen between two substrate molecules at very specific points on the substrate molecules, they need to be held together in the right orientation. So if we look at this anabolic reaction here, so we have this substrate fitting in here and this one here, and the bond forming here between this end of this molecule and that end. So so when these two sit together, this in the right orientation, this bonding becomes easily the bonding happens more easily because of the correct orientation. So in this way, the enzyme lowers the activation energy for the reaction. So here in the slide, we have a little bit about denaturation. So here, this term denaturation is used at extremes of pH and temperatures. So basically what that means is that those weak bonds that hold the tertiary structure in place, like hydrogen bonds, the ionic bonds, they break when the enzyme is denatured. They break, and when they break, there is a loss of the precise three-dimensional tertiary structure of the active site. That's because the polypeptide chain unfolds. And so what happens is the active site is no longer complementary to the substrate. And so what that means is the substrate cannot fit into the active site anymore. And so the enzyme is said to be denatured. And so this would happen, remember, denaturation at extremes of pH and at extremes of temperatures. So before we go into the factors, let's see. So factors affecting enzyme activity. So when we are looking at these factors and how they work, we have two things to bear in mind. First, when the enzyme and the substrate molecules are mixed, they randomly collide, forming enzyme substrate complexes, and that's how enzyme activity happens, as we mentioned. So that means that any factor that influences the collision chances or the frequency at which these collisions happen to form these complexes, so any factor that influences this will affect the rate of the enzyme reaction. And second thing we'll have to bear in mind is the proteins, the enzymes are proteins, and they have globular structure. We'll look at the precise 3D shape of the active site. So anything that will cause harm to this very precise shape, the active site, will affect the enzyme activity, obviously. So 4 of these factors that we're looking at now is substrate concentration, so enzyme concentration, temperature, pH. Excuse me. So let's check them out. So we'll start with substrate concentration. Now, oftentimes with enzymes, we'll get graphs, questions with graphs. So here's a graph, and most commonly the question that will be asked is describe and explain the graph. So then we need to look at the graph and we need to think how many parts are there to the graph to describe. And we need to make sure we describe all the parts and then we explain. So here on the y-axis is enzyme activity on the left. On the x-axis is the substrate concentration. So when we're explaining this graph, we see that there are 2 parts to this graph. So this bit, if you follow my cursor, and then the plateau bit. So this bit here, we can explain it by saying low substrate concentrations. And then this bit here, we can say high substrate concentrations. So obviously, if there are values, we should quote the values. So we'll say at low substrate concentrations, increasing substrate concentration increases enzyme activity. And the explanation is because when we increase substrate concentration, we're increasing the chances of collision of the substrate with the enzyme to form the enzyme substrate complexes. And at high substrate concentrations beyond this bit, at high substrate concentrations, increasing the concentration of substrate does not increase activity any further. I can see the graph plateauing. Why? Is because other factors such as enzyme concentration may become limiting. At high substrate concentrations, all the enzyme active sites are fully saturated at any one moment. So if we go on increasing substrate, this is not going to affect enzyme activity. It's not going to increase it. Okay, so let's look at another
graph here. So this is to do with the effect of enzyme concentration on enzyme activity. Right, so something happened there. Okay, so we can see the graph. We can say that increasing enzyme concentration increases enzyme activity. And again, the reason being that there's increased chances of successful collisions with the substrate, forming more enzyme substrate complexes. Now, as we go on, increasing enzyme concentration, enzyme activity increases. But what is this leveling off? This leveling off has to do with the situation where we're increasing enzyme concentration, but substrate is limiting. So we have insufficient substrate. So that's the time when the graph would be leveling off. Because at this point, substrate is a limiting factor. So the graph starts to level off because enzyme substrate complexes are already forming at the maximal rate. So just one thing to bear in mind when we're looking at these graphs is that we'll look at the y-axis and we see enzyme activity. So when we're explaining the graph, we should be careful not to use terms like the rate of enzyme reaction, because there's no notion of time at all here. So we should not be using terms like rate of enzyme activity or rate of reaction. And we should not be using words like so fast activity, fast enzyme activity, because that is also to do with time. And also another mistake with graphs is people may say there's no effect. But actually, it's not that there's no effect, it's just that there's no further increase in activity. Yeah, there is still a high level. If you look at the enzyme activities, still high. It's just that there's no increase. There's no increase, further increase. So these are things to bear in mind. So let's take a look at another factor that affects enzyme activity. So that's temperature. So here is a graph. And if we had to explain this graph, we would have or describe this graph. There will be three things to explain, describe. There's 1, there's 2, and there's 3. So let's see. So we start by saying at low temperatures below 40 degrees, there is an exponential increase in enzyme activity. Actually, every 10 degree rise will double the reaction rate. Yeah. So why is this? Why is there such an exponential increase? It's because increasing temperature provides more kinetic energy for increasing the collision of enzyme with the substrate to form the enzyme substrate complex. So we mentioned that molecules are constantly moving because they have kinetic energy. If we provide heat, we're actually increasing the kinetic energy for the movement. So we would expect a higher frequency of collision by doing that. And then we say that so 40 degrees seems to be the optimum temperature for the maximum rate of enzyme action. We know that every enzyme would have an optimal temperature for its optimal activity. And what happens beyond the optimum temperature? So at high temperatures above 40 degrees, there is a decline in enzyme activity. Now, whenever we're describing graphs, it's important to give an indication of how of the trend. Is it just a very slow, gentle decline, or is it a sharp or a dramatic decline? So here we can see it is relatively, it is actually a sharp decline. So we would say that there's a sharp decline in enzyme activity above the optimum temperature. And the reason is because of denaturation of the enzymes. So remember, during denaturation, we mentioned that it's the hydrogen and the ionic bonds that break, that hold the tertiary structure together. These bonds break. So that's what we have to say. And because they break, the polypeptide chain unfolds and it loses its complementarity. So the active site of the enzyme loses its complementarity to the substrate. And therefore, the enzyme, as you can see here, can no longer bind the substrate. So the enzyme is denatured. And once the enzyme is denatured because of this high temperature, you can't get back its function by, say, reducing temperature. That doesn't work like that. Yeah, so different enzymes will have different optimum temperatures. So if you go to a hot spring, there will be bacteria in that hot spring. Obviously, their enzymes would be having a higher optimum temperature. It'll be thermostable enzymes. Right, so let's move on to our next factor, which is pH. Now, let's remember what pH is. So pH is a measure of the acidity or alkalinity of a solution. So it indicates the concentration of hydrogen ions relative to water. So a very acidic solution would have a lot of H plus ions, more H plus ions than bases. Bases would have more of OH minus ions. So, so if we look at the graph here, it's like a bell-shaped graph. Here in the middle is the optimum pH. So there's three parts to the graph. There's the optimum pH, and then there's on either side, and there's extremes of pH. So, so the optimum pH brings about the highest enzyme activity because at the optimum pH, the bonding possibilities at the active site are best. They best facilitate formation of an enzyme substrate complex. So what we mean by this is, so these ions, they affect the charges of the R groups of the amino acids. Just a quick recap, we know there's different amino acids making up the polypeptide chain. Some of them have aromatic side groups with no charges, non-polar aliphatic side groups and no charges, polar uncharged side groups. And then we have the R groups, which are positively charged, and the negatively charged ones. So the active side, there will be amino acids with their R groups, and changing concentration of hydrogen ions or OH minus ions would affect these charges. So you can see here, here's an enzyme, and you can see what the active site looks like at optimum pH. It's optimum by bonding possibilities between the substrate and the active site. And here at low pH, we can see there's a repulsion between the substrate and the active site because of the charges. And at high pH, we can see there's negative charges and repulsion again. So optimum pH would be the best. So what happens as we move away from the optimum pH? If we move slightly away, we can see there's a decrease in enzyme activity on either side of the optimum pH. Why? It's because the pH alters the protein tertiary structure and the active site shape. And how it does that is for a disruption of ionic bonds in the tertiary structure. So obviously ionic bonds would be formed by these positively charged, negatively charged R groups of the amino acids. So I'm changing concentration of hydrogen ions or OH minus ions would affect these charges, and hence the ionic bonds in the tertiary structure. Hence, interfering with substrate attachment to form the enzyme substrate complex. And that's what we're seeing here and here. So these are minor changes, but what happens with bigger changes in pH, so really extreme changes of pH, would lead to denaturation. Now, I think one point worth mentioning is that, remember, the term denaturation applies only for extreme changes in pH away from the optimum. And also, if we then say the enzyme is denatured and we bring the pH back to normal, we can get enzyme activity back if we can so the ionic bonds that are disrupted, they can reform if the pH returns to the optimum as it was. So it returns to the optimum. But that's with pH. With temperature, we can't reform these hydrogen bonds and ionic bonds that are disrupted. Right. Okay, so we've looked at pH. So here's a little recap here when we're describing graphs in biology. We're describing trends in the graphs. It's important to be very specific. So here we would say an increase. So this is a steady proportional increase or a decrease. Here we would say, we wouldn't just say increase, we would say exponential increase. For this graph, we wouldn't just say decrease, we would say exponential decrease. So here you'd say exponential increase followed by plateau, exponential increase, peak or optimum, followed by, you wouldn't just say decline, but you would say dramatic decline or sharp decline. Remember, the term rapid, which seems to suggest a notion of time, would be applicable only if this were the rate of enzyme action, or the x-axis were to do with time. So yeah, optimum. So IV here just stands for independent value. So remember, in a graph we have a dependent variable and we have an independent variable. So here on the x-axis we would plot the independent variable. So that's the variable we're changing. And the dependent variable would be on the y-axis. So the dependent variable is that factor that changes, that factor that we're measuring that will change when we change the x-axis values, the independent variable. When we change the independent variable, there's an impact that on the dependent variable. That's why we call it dependent, because the effect that is observed in the dependent variable depends on the independent variable that we're changing. So this was just a little snapshot on enzymes and a few graphs. So if you liked the lesson, then please get in touch, put a like. If you're interested in tutoring lessons, get in touch. My email address is here. It's anushatura@gmail.com. I'll be very happy to help you with your biology. Yes, so I hope you enjoyed today's lesson. Take care, and you're most welcome to get in touch for tutoring lessons. Thank you. So, and see you guys for another lesson next time.